Is my protein of interest present? How can I know? Why, by playing a game of Antibody Where's Waldo? But polyclonal or monoclonal, which way to go? Well, it depends. So antibodies are these little proteins that recognize other specific things. So you could have like an anti-scissors antibody, an anti-pencil antibody. And so they're really useful for being able to see if things are present. So our bodies use them to detect for if foreign invaders are around, and we use them in the lab to see if specific molecules can be found. So from Western blots to histochemistry to flow cytometry, there's all sorts to co-immunoprecipitations, co-IPs, we call them pull down sometimes. Lots and lots of uses of antibodies, and there are some decisions that you have to make when choosing what antibodies to use, how to develop antibodies if you need to do that, that sort of thing. And one of the main things you need to choose is monoclonal versus polyclonal, so let's start there. And although they have a bunch of serious uses and applications, and I'll get into some of this stuff, let's start with a more fun analogy of a game of Where's Waldo.
Say you want to find Waldo, so you send in some detectives to find him. You need to tell these detectives what to look for. And you can tell these detectives different information. So you can give one of these detectives, tell him to look for the puff on Waldo's hat. Another one, look for this stripy part of his elbow. And another, look for the toe of his boot. So these would be like antibodies and the different epitopes that they recognize on an antigen. So an antibody is the little protein that's going to be doing the recognizing. It has this constant region, which is like generic for the animal that it's made in, and it's going to serve as a sort of adapter. There are different types of these constant regions with different types of antibodies like IgG, IgM, etc. Often what we're using is IgG antibodies in the lab, which are these Y-shaped ones. And the constant region at the tips of the constant regions are these variable regions. And these variable regions are going to vary, and they're going to be the place that is actually going to recognize the specific thing. So different variable regions can recognize different places on your antigen. So the antigen is the thing that the antibody binds to. So it has, in the variable region are these antigen binding sites that are going to bind to an epitope on the antigen. So the specific part of the antigen where the antibody is binding is called the epitope. And so this puff of the hat would be an epitope, and this would be another epitope, and this would be another epitope. And so different antibodies are going to recognize different epitopes, and therefore you can have multiple antibodies recognize the same antigen. So we can have lots of different antibodies that are all recognizing the same antigen. So they're all recognizing Waldo, but they're recognizing Waldo in different parts. If an animal encounters Waldo and then starts making antibodies, we'll get more into this, how this happens, but you'll make a mix of antibodies. And so you'll have a mix of detectives going in to see Waldo. And this is like a polyclonal antibody mix. So the clonal is going to be coming from how they're made from individual B cells. We'll get into that in a minute. But the important thing here is that a polyclonal, you're going to have a mix of different antibodies, all of which are recognizing this antigen, but recognizing different parts of him. With a monoclonal antibody, you just have one. So you might have this one, or you might have this one, or you might have this one, but you are going to have multiple copies of these. It's not like you just have a single one. You have lots of copies of them, but it all has the same variable region. You all have recognizing the same epitope. So therefore, we're going to be recognizing just that one part. So we'll get into how these are made in a minute. But let's talk about why you would or wouldn't want to use these potentially in the lab. So with the polyclonal antibodies, you're going to have to worry more about cross-reactivity. So because you're finding multiple sites, if there are other things that have a similar site, then your antibodies might recognize them. So you think that you're finding Waldo, but you're finding someone that's wearing Waldo's hat. We'll show later how this can actually come to our advantage if we put that hat on other things, so we can use like epitope tags to tag proteins if we don't have an antibody against them. But this can be a problem if you have cross-reactivity, in your antibody. On the other hand, however, if you have a monoclonal antibody that's recognizing some conserved feature, then it's going to have cross-reactivity, and then you won't have other antibodies as well that are making yours more specific. So you don't want your antibody to be targeting like a common motif in a protein. And so a motif is just like a recurring thing. So if it happens a lot, if you find the same motif, the same sequence in a bunch of, or the same structure in a bunch of different proteins, and your antibody is recognizing that, that's not going to be very helpful. So you can have cross-reactivity even with a monoclonal antibody. And so this is always something that you need to be aware of and have good controls. An advantage of polyclonal is sensitivity. So because you have more places to bind, you can bind to Waldo in more places, and therefore you can get a stronger signal or response. With the monoclonal, however, you're going to have lower sensitivity because you're only binding to a single site, so you can only have one antibody bound per protein. Although you can then increase the signal with the secondary antibody. So for example, if we're doing a Western blot, so we're taking a mix of proteins and we're separating them by size in a gel, and then we're transferring them to a membrane, and then we're probing that membrane with the antibodies against various proteins. If you have a monoclonal antibody, even if you just have a monoclonal antibody, although the original signal is going to be weaker, when you add this, you can add a secondary antibody typically. So typically you need some sort of way to be able to visualize that the antibody bound. And this is often done with the secondary antibody that binds to the primary antibody in that generic part. And it's also attached, or what we say conjugated, to something detectable. So this could be something like horseradish peroxidase that's going to give off a colored signal, or some sort of fluorescent tag. But because you can have multiple of these secondary antibodies binding per primary antibody, this is going to amplify the signal, even if you only have a monoclonal antibody. We'll get more into this later, but you also have the issue of kind of like consistency between batches if you're doing a polyclonal, and within batches. And as opposed to monoclonal, where you have this single thing, you're going to have no batch to batch variability, or very limited. I mean, you could have like if they purified it worse or better or something, but they should all be like consistent each time you get a batch, but this could be different. In addition, the antibodies that are inside of it, because they can bind with different strengths, and so they might have some really bad binders, some really good binders, some weaker, some binders that have more cross-reactivity, some that are really good. And with monoclonal, you can kind of isolate the really, really good ones that bind really strongly. And so even though you have only one binding site per Waldo, it could be a very strong binding site. The other main consideration when you're choosing a polyclonal versus a monoclonal antibody has to do with cost and feasibility and that sort of thing. And so to understand this, we need to understand better how these antibodies are made. And so let's go into some of the ways that antibodies are made in the lab. Starting with the ways that antibodies are actually made in the body. So in our body, in the body of animals, you have these B cells. And so this is a type of immune cell, and they have the instructions for making these antibodies, and they kind of edit the instructions. And so it's happening just in these individual B cells are making these random choices in how they're editing the DNA, editing the genes that have the instructions for these antibodies. And this leads it so that each individual B cell is going to make a single antibody. And it has a single variable region. And then these are going to go out, and if they find an antigen, so if they find something like something foreign that isn't present in our body, then they're going to get selected for. And when they get selected for, then your body is going to make more copies of it, or the animal body is going to make more copies of it. And it's going to expand. It's going to like clonally expand. So basically you're going to make lots of clones of this cell. So this original cell is going to make lots of copies of that cell, as well as lots of copies of antibodies. And so you start with the single cell, but then you expand it. So you get lots and lots and lots of cells making this specific antibody. And in the body, what's going to happen is you're going to have different B cells recognizing different things or different parts of the same thing, so like different epitopes, and they're going to be making different antibodies. So what happens if you want to get an antibody against a protein of interest? Typically in the lab, at least, we'll talk more about later about some of the animal-free strategies and some of the later technologies. But this animal production is still used quite a lot. What the basic overview is that you inject the animal with the antigen, so with the thing that you want them to recognize. Often you do this with some sort of adjuvant, so like a chemical that's going to tell them, okay, your immune system, wake up, this thing is foreign. When you introduce this antigen, then the antibody, then the B cells in that animal are going to start, their B cells are going to have the opportunity to see if they can bind. And if they bind, then, and if they don't bind to anything in the animal, then they're going to, like any self things, then they're going to get chosen, they're going to expand, and then you're going to get lots of these antibodies made. Some of these antibodies are going to get secreted into the blood. And so if we capture that blood and then we purify out the antibodies that bind to the thing of interest, then we're going to get a polyclonal antibody mixture. So we can just take, you can just take some like beads or something that are attached to the antigen that you want the antibodies to recognize, and then you can purify out, so capture the ones that bind, and then purify them further and stuff, and have a polyclonal antibody mix. But you're getting all of the antibodies that bound to that thing. So you're getting all of these ones that recognize all of these different epitopes and are coming from lots of different B cells. If you want to isolate a particular one, then you want a monoclonal antibody. Now you want to do is isolate the actual B cell that made that antibody and get it to make a lot more. And so this is a lot more difficult of a process. So with polyclonal antibodies, this is typically done in like bigger animals, and it is done, so like rabbits, goats, things like that. And what happens with these is that you're going to, each time that you buy a new batch, basically, they use these bigger animals because they're collecting them from the blood. And so these animals have more blood, they're going to have more antibodies that you can collect. But it's going to be like a limited source, because you have to, if you want more, you have to inject another animal, or you have to wait longer, that sort of thing. And each time that you get a batch is going to be different because it's going to be coming from like a different animal or that sort of thing. And so you're going to have more batch to batch variability, which can be a problem. With the monoclonal antibody, here you're going to have kind of like a limitless source, but the big cost comes from isolating that source. But once you have that source isolated, now you can make lots of the same antibody. So you get the consistency and you don't have to worry about running out of it and having to go to a new batch. Well, I mean, they could have a new batch, but it would be the same thing. So how do these antibodies actually how do this monoclonal antibody technology work? There are some different ways. So conventionally, like the old school method that's still done in some cases is this like hybridoma technique. So basically you take a B cell from the immunized animal, and this is going to be good at growing and making the antibody you want, but not growing outside of the body. And then you fuse it to a myeloma cell. So this is a type of cancer cell. And so the cells are really good at growing outside of the body and growing lots and lots and lots, but they can't make antibody. But when you fuse them together, you get this hybrid called a hybridoma. And now this can grow and grow and grow, and it can make antibody. And since you only have one original B cell fusing with the myeloma cell, you only have the instructions for making one specific antibody. And so you're only going to make that one specific antibody. Alternatively, what's getting done more and more frequently is that you actually take those B cells and you sequence to find the information, find the genetic instructions for actually making that antibody. And then you could stick those instructions into expression cells, so into like CHO cells or bacteria cells, different types of cell lines, and you can actually make this just in cell culture. But in these cases, we need to be able to isolate those B cells. And so there are different ways to isolate the B cells. These days, they have like cell sorter, a bunch of cell sorter technologies where like microfluidic things, you flow things and then you stick the B cells into individual wells and get them to make antibodies, various things like that. The basic principle is that you only want to select the B cells that are actually making the antibody of interest. So you can do it in something like this, where you have some sort of fluorescent signal that's going to be given off if you have an antibody, a fluorescently labeled antibody that matches the antibody that's made, that matches the thing that you want it to recognize. And then you can select out those B cells. A common way to see if an antibody is binding to something of interest is an ELISA. So this is like where we're talking about with a Western blot, and that you have this sort of like secondary antibody thing going on. In this case, however, you have your antigen bound to the well. So instead of having it like potentially on a the membrane from a Western blot, you have it definitely on all of these wells of this plate. Then you have put the antibody on top, or so you could put, say, the liquid that the B cell was growing in on top. If it has the antibody that recognizes that antigen, it's going to bind. You can wash off other stuff and then add a secondary antibody that has the label and see if you get detection. And so then you would then say, okay, let's take that B cell and move forward. So there are different sorts of techniques. In terms of the animal sources, so often with if you're isolating B cells, then that's often done in like mice. But for polyclonal, where you want more of the blood, you use bigger animals like rabbit and goat and things like that. But so those animals have antibodies that are pretty similar to ours. And if we want like humanized antibodies, such as for use in the lab, for use in medicine, such as like the monoclonals that you talk about with treating COVID-19, in those cases, you want like humanized antibodies so that our bodies don't recognize them as foreign, and they actually have like specific mice that are engineered to have like make human antibodies. So have the human constant region and stuff. Pretty cool stuff. But so all these antibodies are kind of bigger and bulkier than the antibodies that some of the animals have, such as llamas. They have these cool like heavy chain only antibodies. And so the key thing is that you can make this part of this like nanobody that is really small. And this can be good because it can recognize really small parts of proteins and stuff and get like into crevices. So these can have some cool uses, and there's more like nanobodies being used and that sort of thing. So that's an area to keep an eye on, as well as technologies that don't use animals at all. So one of the techniques that you can use that I've talked about before is phage display, which is this cool technique where you take this virus that infects bacteria, so this bacteriophage or phage. These phage have these coat proteins that are going to stick out, and you can actually fuse another protein or a part of a protein onto this coat protein. And then this is going to be displayed from the surface of this phage. So what you can do is you can actually take parts of the antibody and fuse them to that phage protein. And now they're going to stick off of this phage. You can then make a library of millions of different phage clones where they're each expressing a different antibody. So you can mix and match all of those different constant, and you can mix and match all of those different variable regions in order to find different combinations that are going to match. When you do this, then you let these phages infect bacteria and make lots of the phages. They're going to bind to then you see if they can bind to the antigen that you want, wash off ones that don't. Then you elute them, so you unstick the specific ones that stayed on with your washes, the ones that bound specifically to these, and then you amplify them, so you let them infect more bacteria to make more of them, and then you can go and sequence them and actually see the DNA that was in those ones that bound. And you can do this in multiple cycles, and you can do things like mutagenesis to help mature them and increase the binding affinity. But in this case, you're basically skipping the animal and you're doing this all in this phage display format. And so some really cool stuff, and definitely an area that is growing in use, as well as some other technologies for antibody design and manufacture and that sort of thing. And these technologies are important, not just because of medical uses, such as making antibodies that we hear about in the news all the time. Speaking of which, when you talk about like monoclonal antibodies and antibody cocktails, and versus polyclonal is going to be is different from an antibody cocktail, but antibody cocktails are like a mix of monoclonals. So basically you can make specific monoclonal antibodies that are really good at binding what you want. But you have the possibility of like viral escape if we're talking about an antiviral antibody. So if one antibody binds, but then that part of that epitope that it binds changes, then you're not going to bind anymore. But if you have another monoclonal antibody that's going to bind to another part of the protein, and that part doesn't change, then you still have something that works. And so with an antibody cocktail, you're mixing different monoclonals, but each of those were monoclonal. So you isolated the B cell, and then you made or you somehow found the instructions for making a good antibody with phage display or something like that. And then you make lots of copies of it. And then but you're mixing different monoclonals, but this is not the same as a polyclonal. A polyclonal is just like whatever the animal makes and that sort of thing that happens to bind. And so it's coming from lots and lots of different B cells. In the lab, there are lots of different uses for antibodies. So we've talked about the Western blot. There are other things that you can do, like a co-immunoprecipitation, a co-IP, sometimes called like a pull down. So the basic idea is that you have some beads, and then they're attached to an antibody that binds to something that you're looking for. And then you can mix those beads with a mix of things that might have what you're looking for. And then you see what binds, and you're going to bind what you were looking for. And you're also going to bind whatever what you were looking for was bound to. And so then you can then look and see what it was bound to, such as by doing a Western blot to see what was there, see if a specific partner was there, or by doing like some sort of mass spec or something to see what it was actually bound to. If you can also do something similar where you wash it off better, and you can use it to purify a protein. Of course, this technique can only work if you have an antibody against the thing that you're working with. And as we just went into, this can be a long, expensive process to make an antibody that's specific for a protein of interest. And often these ones that are like specific for proteins of interest can have a lot of cross-reactivity. So it's really important that you have good controls and that sort of thing. And not all antibodies are created equal. But if you have a protein that doesn't have an antibody of interest against it, all hope is not lost. And so if you're using some sort of recombinant technology where you're actually like sticking the instructions for making the protein into cells and getting them to make them for you. So we call this recombinant because you're recombining the gene of your interest with something that's going to allow you to get it into cells and make it. Alternatively, you can do this in cells with some like genetic editing, where you're actually modifying the gene inside of the cells. In either case, what you can do is you can add a little bit extra to the end of your gene, and this is going to serve as an epitope for the antibody. So remember that the antibody is only recognizing specific features, such as the puff of the hat. And so you can use an antibody that recognizes the puff of the hat, and then you put that puff of the hat on top of your protein, attached to your protein, and now you can use an antibody against that. So you can add an epitope tag. Common ones include like HA, flag, MIC, V5, 6X. So these are just really short sequences that you put on the end of your protein by adding a little extra genetic information onto the end so that it gets made as long with your protein as part of your protein, like on the end. And then you can use this for various things. This can be really helpful, and because it's this short little tag that gets recognized, there's less of a concern about using it for different purposes. So there's sometimes when you want to use an antibody to recognize proteins in their native or their folded state. There are other times when you want to recognize them in their denatured state, so like their unfolded state. So when you're doing a Western blot, for example, you're denaturing the proteins with SDS page. So you're unfolding them and then you're sending them traveling through a gel. And so when you go to probe, you're going to be probing for the unfolded protein. If the antibody was recognizing part of the folded protein, if it's recognizing like a conformational isotope, so it basically requires the protein to the shape of the protein. So if say like you have a binding site that's made up of parts from this part of the protein and parts from this part of the protein, and so they're not like linearly connected, but they're actually like spatially connected, then that wouldn't be recognized by the antibody. But if you just have like a short little tag as like linear epitope, then you would still be able to recognize in both cases, as long as it's not hidden by the way your protein's folding. So it could still be hidden, but there's less of a risk. The key thing is that you need to make sure that the antibodies that you're buying are validated for the purposes that you're going to use them for. So for example, if you have something that recognizes a linear sequence that is somewhere in the protein that's normally hidden by folding, then that would work for a Western blot, but it wouldn't work if you were doing something where you're trying to like stain the protein inside of cells, so some sort of like immunohistochemistry or flow cytometry, where you use antibodies against like some surface protein or something, and then you're sorting them based on their fluorescence, or you're staining them based on their fluorescence and sorting. I don't do that sort of thing, but you're recognizing that proteins, you're recognizing proteins in their native state, and so you need to make sure that the antibodies you're using recognize them in that native state. And so all the information should be findable, hopefully on the like the website for the antibodies that you're buying.
So I've talked mostly in terms of different proteins, but antibodies can also recognize things other than proteins, as well as modifications to proteins. So an antigen does not have to be a protein, it just commonly is, especially when we're talking about uses in the lab.
So those are the main things I want to say about antibodies in terms of their lab usage and production and all of that stuff. So I hope that was helpful.